Okay, you're good to go.
All right, so the first question is epigenetic inheritance, and we're doing the Engelmann syndrome, and we know this is paternally inherited alleles. The imprint silences the paternally inherited alleles, so that means that all the alleles you get from your father are silenced. So we can pretty much pick the mother's alleles and separate them, and that'll be our phenotypes. So for our first phenotype A, we have one non-Engelmann and one Engelmann. The second one, same thing. So our mother is heterozygous, or I mean, sorry, for the second part, the genotypes are 1AA to 1AA homozygous, and then 2 heterozygous, and then 1 homozygous recessive. The second question is you do a cross with a homozygous recessive female, so that's going to be your phenotype. So all phenotypes are recessive and have Engelmann. Your genotypes are different. One is you have a ratio of 1 heterozygous and then 1 recessive homozygous. For C, whenever your XICS has been deleted from your X chromosomes, no Barr bodies will be formed because you need at least 2 to form a Barr body. So the answer is 0. And for D, T6 is the one that stops exist from making a Barr body out of the X chromosome. So if you don't have any T6, they're all going to form Barr bodies. So you'd have 2 Barr bodies formed.
Okay, so for number 2, we had the fill in the blank section of the group problems. And for A, we got SRY is an example of holoandric gene, of a holoandric gene. For B, we got MIC2 is an example of a gene found on both the X and Y chromosome. C, for this one, it says a cross is made between a IGF2 negative male mouse and an IGF2 negative female mouse. What are the phenotypes of the offspring? And for this, we went ahead and we did a Punnett square right there, and we got half would be normal and half would be dwarf. For D, we got sex influence inheritance. So it says in the sex influence pattern of inheritance, an allele can be less in another sex. And then for E, we got genomic imprinting is a epigenetic process that occurs during gamete formation, while dosage compensation is an epigenetic process that occurs during embryogenesis. For F, we got the question is, one of the topics discussed in lesson 2 involved the formation of a double-stranded RNA molecule. What 2 molecules from the 2 halves of this double-stranded RNA? And we got X ist cells and T6 cells, and T6, Xis and T6. For G, when reciprocal crosses give identical results, the alleles that control the trait in question are located on, and we got autosomal. And then when reciprocal crosses give different results, the alleles that control the trait in question are located on the X gene.
For question number 3, it is the OTD, the deficiency one, which is an X-linked recessive disorder. So for A, it asks what's the genotype of the woman's father. And because he has the gene and it's X-linked recessive, he has to have a recessive on the one X gene he has and then the Y gene, because the Y gene doesn't play a part in the deficiency. And for the mother, she is homozygous, is what we know, and unaffected. And if she was affected, she would have to have both recessive on her genes. So I know that it's the father is X recessive Y and the mother is dominant for both. For B, if the woman has children with a man who does not have OTD, what is the chance that her first child will be a son with OTD? We did a cross between, let me see, find this. Right, this one, I don't know if anybody's seen it. Anyways, we did the cross between the female, so who is X, who is heterozygous, and the male who is homozygous dominant, and it gives us 50% chance between the two males. If it's between all the children, it would be 25% that they would have OTD. For C, identify the genotype of a male with whom the woman could produce a daughter with OTD. The male would have to be recessive. And to check that, we just did another cross. It's right here. And checking it, it came out to be right. Consider the cross in part C of this question. What are the expected genotypes and phenotypes of the offspring? The genotypes, we have the females, one is heterozygous and one is homozygous recessive. The males, one is homozygous dominant and one is homozygous recessive. And then for the phenotypes, we have one normal female to one OTD female and one normal male to one OTD male. And that was just from the same test cross that we did in number C.
Okay. So for number 4, we had to do test or normal crosses for all of them, but this is with goats, with or without beards. So for the first one, we did Punnett square, and it told us that the males, 3 had beards and 1 did not have a beard. So from that, we had to figure out the offspring, and from it, figured that both the male and female have the same genotypes, which is big B, little b, and then big B, little b again. And that creates the 3-to-1 ratio. And then that's for A. And then D is actually the same, but since the female, we're talking about female offspring, so the dominance switches to the little b versus the big B. And so the female is big B, little b, and the male is big B, little b as well, as you can see here. And then for C, we had 1 bearded offspring and 1 not bearded offspring for females, which would be 2 and 2. And so I had to remember in this one that the female is the little b is dominant instead of the big B. So did the Punnett square, and we've got 2 that are bearded because they are both have the big B's in them, the little B's in them, and then 2 that are not because they don't have the dominant little B. And then for number D, we have the same thing, but it's in male offspring.
I know, stay on top of stuff.
So we have the female is little b, and the male, sorry, switch that. The male is big B, big B. The male is big little b, and the female is big B, little b. And then in that case, the big B now makes the males have beards, whereas the little b does not. And then that explains the lack of beards for number 4. Okay, so for number 5, we're talking about X-linked inheritance and reciprocal crosses. So for A, they're wanting us to cross a orange male cat to a tortoiseshell female cat. So when you cross them, we did our little Punnett square right up at the top. And so we found that with that cross, there would be a quarter of them would be orange females with the genotype little b. A quarter of them would be tortoiseshell females with the big B and a little b. A quarter would be black males with a big B and a Y. And then a quarter would be orange male with a big B and a little b and a Y. So for B, when it's asking what the genotype of a male tortoiseshell cat would be, that would mean that they'd have to inherit an extra Y chromosome, so their genotype would be big B, little b, Y. For C, talking about the scalloped phenotypes for wings on fruit flies, we know that the X-linked recessive, the scallop is an X-linked recessive mutation. And so when you do a reciprocal cross, we did the female being homozygous recessive and the male being dominant. And so when you do that, you get a ratio of 1-to-1 of, let me say that again. So you get 2 unaffected females for every 2 males with scalloped wings. And I have my work here to show that right there. And then for D, doing a autosomal recessive inheritance, there would be 1 parent that's homozygous dominant and 1 parent that's homozygous recessive. And so that means that all offspring will be heterozygous. And so all of them would be unaffected. So they wouldn't have to have the scalloped wings. So that is number 5.